are very rich by active compounds and many other nutrients such as fibers and proteins. Therefore, this is one of the best strategy to use agriculture byproduct waste for feeding our veterinary animals. It will provide high nutritive value as well as quality of meat and milk as well. It will provide us economical advantages as well as industrial advantages, because it will reduce the cost of disposing of agriculture byproduct waste when we use this agriculture in the feeding of animals. Therefore, this study is designed to use hazelnut that have about 50 to 55% of dry matter content as well as 15 to 21% of energy. We know that in the hazelnut production industry, after roasting, 50 to 70% of the hazelnut is wasted, and this 50 to 70% of hazelnut waste can be used in feeding our ruminants and other veterinary species. Therefore, there is a great need of time to use these agriculture waste in the production of ruminants and other animals. There is a research gap present in the data, because there is no such study present till yet for utilizing hazelnut or its hazelnut skin for dairy animal feed. Therefore, this study is designed to use hazelnut skin for feeding bullocks of Italy, and to check the meat quality, their quantity, and sensory profile, their oxidative stress profile, and lipid peroxidation profile. Keeping in view, here are three objectives of my research. First objective is to investigate the effect of hazelnut skin on the oxidation, microbiota profile, immunity, as well as serum protein expression of bullocks that are fed with hazelnut skin. Second objective is to identify that either hazelnut feed have any effect on physical, chemical parameters, and either it also affect stability of meat and its density profile of meat. Third objective is to check sensorial profile and nutraceutical properties of meat. Well, this is my research design. First of all, I will collect about 20 samples of bullocks for hazelnut feeding, and 10 samples as a control that are fed on other diet other than hazelnut. Well, to the 20 sample, we will feed hazelnut skin along with chopped alpha hay, and we will continue this experiment almost for 60 days. And after 60 days, I will collect blood samples and fecal samples. Blood samples will be used for the analysis, biochemical analysis, that will include total protein, albumin, globulin, urea, creatinine, and alkaline phosphatase analysis, and other immunological parameters. Fecal microbiota sample will be collected for cytokinin expression. After 60 days, after collection of fecal samples as well as blood samples, we will slaughter these experimental bullocks, and then we will check other parameters of the meat, such as sensorial profile, as I have told you before. Sorry. Just a minute. Let me show you my slide. Okay. After slaughtering the animals that we treated with hazelnut feed, we will get them slaughtered, and we will check the quality of meat by comparing water holding capacity of control bullocks meat and hazelnut feeded bullocks meat. We will also check Warner Ballinger shear force and also check the pH of the meat. Second, we will analyze lipid peroxidation level by using TBARS, thiobarbituric acid reactive substance species, that is a good marker of lipid peroxidation in the meat. Other than this, I will also check hydroperoxide levels in the meat and protein carbonyl that are present in the meat. Other than this, I will, as I told you before, that I will check I will collect cacal biosample after slaughtering. I will check the microbial profile of meat, that either any pathogenic bacteria are present in the meat, or they are improved by the hazelnut feed, or there is no difference between control population and our experimental population. I will also check some gut cytokinin protein expression, and nutraceutical profile of meat will also be checked, and carotenoid analysis, vitamin E, vitamin A, and this will be checked for the nutraceutical profile of meat, and at the end sensory profile will be checked by sweetness, juiciness, its taste, and tenderness of meat, and it will be checked according to the British International Standards. We will take almost 15 individual assessors, and they will check sensory profile of meat, and they will tell us about sensory analysis. These are expected results of my research. We expect that our experimental population of bulls meat will have high meat quality, and the quality of meat will also increase the shelf life, and it will reduce oxidation damage in the experimental bullocks. We also expect that the pathogenic microbiota will decrease in the hazelnut feeded bullocks as compared to control bullocks, and there will be low oxidative stress in the meat as compared to control population. We also expect that this feed will improve sensory profile of meat by increasing juiciness, tenderness, and its color of meat. At the last, we also expect that the polyunsaturated fatty acid profile will be high in the treatment group as compared to control group. That's all from my side.
Thank you, Shamsa. And you can stop the sharing of the screen if you want.
So, Professor, actually, nowadays I'm suffering with pneumonia, so I'm facing breath.
Don't worry.
So,
don't worry, it's not a problem at all.
okay.
I got one question concerning your project, and
yes,
one question of a more general overview concerning the general idea about the project. Why did you indicate the 60 days for the sampling? And this is the first question. So how did you get there, and how did you thought about the dosage of 220 grams of ethanol skin to be included? And the second question is if you could please imagine or explain the impact of the project, and who can be identified as possible stakeholder, and how would you plan to involve them in the activities?
Well, first question is about the sampling number. Actually, I selected 20 samples as an experimental group and then for control group. Actually, I selected 20 samples because, as I told you before, I have to slaughter these animals to get meat and to check the nutraceutical profile, sensory profile, lipid peroxidation profile of meat. So, because this is an invasive type of study, I have to slaughter these animals. Therefore, I selected only 20 species to you can say to meet the criteria of any institute to sacrifice animals. Therefore, I selected only 20 species. And as far as 60 days is concerned, it's about 2 months, and I selected these 60 days because as I studied in literature, most of the studies that use any agriculture bioproducts, they use about 30 to 40 days after time period to feed the livestock and then check milk profile and like studies. Therefore, keeping in view this literature studies, I selected 60 days instead of 40 days, because in the literature mostly in the literature 30 to 40 days are present. But I increased this number to 60 days to better identify the meat quality, because I think that blood profile can be can be changed very rapidly by changing feed to animals, whereas meat profile, sensory profile, and other nutraceutical properties need some time to store these bioactive compounds and nutrients inside meat. Therefore, I prolong this period to 60 days. And as far as stakeholders are concerned, I think that every person needs high sensory profile meat that is fresh, that have high juices, that have high tenderness level, and this attract the customers. And when we will find any strategy, any feedstock that can improve sensory profile of meat and the nutraceutical profile of meat, and then it will be mentioned over the meat that this meat contains these profiles and these nutrients, then customer will be more attracted. And all the industries, dairy industry stakeholders definitely attracted toward this research, and they will try to add this research in their domain to better sell their products. Therefore, dairy industry is at more benefit by this research.
Try to imagine a wide angle. So you basically identified the consumer as possible stakeholder, which is for sure correct, and the meat industry, not the dairy one. Maybe the meat one is more interested in this kind of project with Budlock. Going back in the productive chain, who do you think can be involved in the activity? So taking into account that this is an industrial byproduct, how and why would you like to involve other stakeholders? Because to perform a project, we need to involve different professionalities. So which would you identify before the consumer and the dairy industry?
Yes.
To be interested in this kind of project, because that is part to begin the activity.
Oh yes, I understand. I think you mean that before going to consumers, you are asking that before going to consumer, which stakeholder will involve, right?
Yeah, you have to imagine the whole productive chain to have a wide overview of the issue we are facing together, and imagine for each phase the different possible stakeholders to involve.
I understand your question. This question is very interesting as I am from Pakistan, and Pakistan is such as dependent economy on agriculture and livestock production. And if I talk about the perspective of Pakistan, there is a great need of time to improve livestock production and their meat quality as well as dairy products, right? So if I talk about Pakistan, there is a great need of time to improve meat quality and all the ruminant animals, because they are consumed in a high quantity in Pakistan. So these type of stakeholders and government bodies in Pakistan and some other private organizations will also involve as a stakeholder in this study.
Okay, just one last question concerning the feed technology, because basically we are trying to include a new ingredient in animal feed, so we have to consider even that part. And what do you think could be the effect of this kind of new ingredient in the feed technology, and what kind of action would you perform to confer this kind of effects?
Sorry, Professor, what you said?
What kind of effects do you expect to be observed in the feed? So talking about the feed technology, and what kind of action do you perform on the feed to confirm your expectation, your results, your expected results?
All right. As I told you before, that the hazelnut skin, about 70% of hazelnut skin is wasted after roasting in the industry, and this hazelnut skin is a major proportion of waste product of industry. When we get this hazelnut skin from any hazelnut feed industry, and we will collect this sample, and then we will check all the nutrient properties and all the bioactive compounds present in this buy a product that is wasted by industry, we will check the profile of this hazelnut skin, and as I have told you before, this hazelnut skin profile is high in fiber content as well as protein content, whereas the water content in the hazelnut skin is very low, right? So there is no need to you can say there is no need to put effort or any energy to dry this matter, to store this matter for a longer period of time, whereas the bioactive profile, bioactive compound profile, proteins and fiber content is very high, and this high protein and fiber content definitely increase the tenderness as well as the freshness of milk, and it will also increase the sensory profile. Therefore, when I collect sample from industries, I will again check all the bioactive compounds and nutrients present in this stock. What I tell you before is all from literature. What I've studied in the literature, that there is high content of protein and fiber present. Other than this, I will check this hazelnut skin bioactive compound content and nutrients before starting experiment.
Actually, we have, and this is just a comment, and what you said is quite correct. There is something missing, that the problem of this kind of ingredient is the high quantity of lipid content. That's why we have to take care of the storage, and even if it's dry, we want to preserve the antioxidant part to be used by the animal and not to preserve the lipid content of the skins. So we have to take care of that and be sure that adding these ingredients to the bullock diet, the lipid content will not increase too much, because this can be detrimental to the animal performances. So this is something we have to deal with when planning a precise diet for this kind of bullock. But thank you. You've been quite efficient, so thank you very much.
Thank you for having me. Okay. Can I leave now?
Yes. Yep.
Okay.
Thank you very much. The committee is okay. Okay. So thank you.
Okay, thank you.
We can move to the last candidate, Elena Diaz Vicuña.
Yes, can you hear me?
Yep. You are Italian, so if you want to conduct the talk in Italian, you can, but that's up to you.
Okay. Well, actually, I studied for the presentation in English, so if for you it's okay, I can conduct.
Oh, that's better for us. That's better for us.
Okay, then.
So.
Before to start, Shamsa, can you please stop the screen sharing? Okay, thank you very much. Elena, as for the others, I ask you to briefly describe the project, and then we start with some questions.
Okay, I will briefly share my. Let's see if I can do this. I just want to share the screen. Should be this one. Can you see anything? No.
Not yet.
Wait. Excuse me. Okay. Now it should be possible to see my project. I didn't make a PowerPoint, so I'm going to simply present the PDF that I uploaded when I presented the demand. So my project is the hazelnut industrial byproduct inclusion in Piedmontese beef cattle livestock system. To give a little bit of context, this project develops from a necessity that is, from a very general and wide point of view, the necessity to solve and to try to contrast a growing issue that is the world hunger. Obviously, this is just a part of a much more complex picture that needs to be, that in order to be perfectly understood, needs to contain many more factors, such as overpopulation, global changing, climate changing, as well as land abuse and the feed and food competition. Therefore, in order to solve these growing issues, many solutions can be applied, and most of them can be actually put under a single name, a single action, that is the switch toward a much more circular economy. Therefore, in order to put this idea into practice, it has been many solutions are now currently being investigated in the literature, and as for now, the solution that has been identified, that can be identified as most effective, is the application of co-product / byproducts that can be obtained from the food production supply chain as animal feed. In particular, the hypothesis driving this project is the possibility to use hazelnut skins as feed for beef cattle. Therefore, hazelnut skins are defined as an agro-industrial co-product that are obtained from the hazelnut roasting, and that, as for now, is characterized by the absence of any economical value. They're basically disposed as waste, and therefore and they are characterized by a very interesting nutritional profile. They present a very high value of fiber and also total lipids, in particular unsaturated fatty acid, that are currently actually being requested in the current market demand as for the animal-derived products. They are also very rich in bioactive principles, such as tocopherols, as well as phenolic compounds that are perfectly known for their antioxidant as well as antimicrobial properties. And moreover, beside this very important nutritional part, there's also an economical side that needs to also be addressed, because the hazelnut industry is pretty relevant in Italy right now. The latest data reports that Italy ranks as the second highest country as for the export of hazelnuts, the first only being Turkey. And also from a qualitative point of view, it is reported that Italian hazelnuts are actually preferred abroad. Moreover, we are also the homeland of some of the main confectionery plants, starting from Ferrero to Perugina, etc. Therefore, this is the state of the art of what we basically know about hazelnuts and hazelnut skins, and there is currently a gap of knowledge in what we know, that is the possibility of inclusion of hazelnut skins in the livestock feeding sector, in particular in the beef cattle. Therefore, in order to answer this gap of knowledge, and also in order to produce more data, new data that can be compared to the already existing ones, I have decided to suggest this project. The objectives, therefore, would be to work on an environmental area, on a nutritional area, and also on an economical area. As for the environmental area, the objectives would be to reduce the impact from the hazelnut industry, but also from the livestock sector, because this would allow us to work on one of the main currently hot topics of the cattle sector, that is the feed area. At the same time, this project would also aim at improving the animals' welfare as well as the derived products, since the starting point would be to feed much more safer and also nutritionally higher products compared to the currently used ones, and also to finally involve also the consumers. The project also therefore would also explore a part of consumer science through a survey that would be conducted both face-to-face and also through internet. So I would suggest, moving to materials and methods, I would suggest a multidisciplinary study aimed at investigating three main areas. First of all, the hazelnut skins through a characterization of the product. Second of all, the animals, so through an in vivo trial. And lastly, the product, the animal product itself, through a panel of analysis. To start with the analysis of the hazelnut skins, I would suggest to determine the chemical profile as well as the energetic value, and while focusing on the fatty acid composition, which is which interest has been growing in the consumer's opinion, in the phenolic content and for the antimicrobial properties. Because the literature already presents some data, but they are not all. There are some varieties, so it would be interesting to perform some preliminary analysis. Moving on to the in vivo trial, I would have suggested to divide the wind counts into groups, a control one and a treated one, and obviously the control would be fed only conventional concentrate, whereas the treated one would receive a mixture of the hazelnut skin and the same concentrate. I would propose a productive cycle of 13 months, at the end of which during which I would record live performances, such as related to both the welfare area as well as the productive area, such as for the welfare area, serum collection, blood collection, as for the immunity, in order to evaluate the immunity. At the same time, also performances such as body weight gain, and also the feed conversions efficiency, etc. Moving on to finally the meat analysis, I would plan to define the physical, chemical, nutritional traits, while once again focusing on the antioxidant capacity, because this might be a very important point of focus for the product itself. I would also explore the shelf life through microbiological exams, and finally, I would, as anticipated, I would also explore the consumer's opinion on this type of product through surveys, in order to evaluate also their opinion on this type of meat, of this type of produced meat. To conclude, the expected results would be an overall increasing of the health and welfare of animals, humans, and of the environment. Starting from the environment, as aforementioned, I would expect an improving of its state due to the reducing of waste on one side, and at the same time due to the reduction of the feed versus food competition, and to the carbon footprint related to the livestock, etc. As for the animals and human health, as the One Health principle already states, they are strictly interconnected. The idea is that through feeding a much safer, a higher quality type of feed to the animals, it is expected to find much healthier animals, and therefore it is expected to create a much safer product for the human consumption. This would mean, in conclusion, another step toward a much more circular economy, and therefore an overall healthier future. And that's all. Thank you.
Thank you. And as for the other candidate, I'm going to ask you something about the project and one question about to provide a wider overview. So for the first question about the project, you identified a very precise productive chain, which is the Piedmontese bullocks. And if you could please explain why and which kind of advantages the Piedmontese bullock productive chain can have using this kind of product. While the other one, you somehow well described the scenario in which we are moving. There is something missing, because there is not only the feed food competition, but we are also dealing with another very hot topic, which is the antimicrobial resistance issue, which is one of the main One Health topic we face here in the department. So can you imagine somehow positive effects this kind of diet can exert to fight that kind of issue, so to fight the antimicrobial resistance. So there are basically two different questions. You can take your time.
Okay. So starting from the first question related to the Piedmontese beef cattle. Well, I decided to focus on this type of breed for many reasons. First of all, the Piedmontese beef cattle, the Piedmontese breed is currently one of the least reported. Basically, I couldn't find proper data in the literature. Therefore, I thought that this could have represented an important way to fill the current knowledge gap. Secondly, I also the Piedmontese cattle, as for now, is one of the most standardized type of breed. It has been genetically selected for many years, and for now might represent one of the. This would allow much more repeatability, a much more a higher robustness of the test, because I also thought that by picking another type of breed, a much more rustic one, some data might have been different due to bias related to the breed itself, to the heterogeneity of the breed itself. Therefore, I thought that also this might allow a higher repeatability. And moreover, well, the Piedmontese breed is also one of the most important breeds in Italy. And the study that this project is aimed at providing results that can be applied in the reality, in our reality, the Italian reality. Therefore, I thought that this could have also helped in creating a much more robust study, and also providing data that could be applied much more easily for other studies. I don't remember if you asked me something else before.
Now you can move to the other question.
Okay. As for the other question, yes, the antimicrobial resistance is definitely a very hot topic, and as the expectations of the application of this type of product in the beef nutrition would be to determine on one side an overall increase, as aforementioned, an increase of the overall health of the animal, and this would allow to better contrast any type of illnesses, in particular, for example, illnesses due to bacterial infections, thus allowing to reduce the use of antimicrobial substances. And that's why I previously stated that the use of this type of hazelnut skins could potentially lead to the creation of a safer product, because by creating healthier animals, much more robust animals, and that's why I also propose to control through blood samples and the level of immunity of the animals. This would allow to create a product that contains less chemical, that has been in contact with a reduced number of chemicals, and thus creating, as aforementioned, a safer product, and at the same time reducing the possibility of creating implementing the antimicrobial resistance. Moreover, the same antimicrobial properties due to the presence of phenolic compounds and tocopherols might also allow to reduce the bacterial growth in the feet at the beginning, thus allowing once again to reduce the use of substances, also to control the product shelf life.
Okay. For me, it's okay. I don't know if the commission has some adjunctive question.
Okay, thank you very much, Elena.
No, thanks.
That's okay. Thank you very much. So I thanks to all the candidates, and I pleased to disconnect from the room. We will discuss and we'll fill the form and all the documents, and you'll have notice about the results from directly on the website. And thank you very much to all, and I please Claudio and Annalisa to stay connected to discuss about. Thank you very much.
Thank you very much.
Excuse me, can you hear me?
Yes.
Okay, I just hear that you are going to close. You didn't ask me for the interview. Am I right?
No, because we have just 3 candidates for the oral part of the talk. You are... So what's your name?
I am Ziaullah.
Sorry, could you switch on the webcam?
Yes. Can you see me?
Okay, yes.
Okay. I am not in the interview list,
You are not in the list, I'm sorry.
but I got an email that you are invited for the interview. I don't know.
Maybe it depends by the point you reached at the title evaluation. Maybe it was not clear to them. I don't know.
Yeah.
Oh, okay.
The advice is that today there is the colloquium, but not by person. There is a graduatory on the website, and the people that pass the exam of the title
Okay.
are Abbas Mumastar, Kanval Samsa, and Vikuna Elena.
Okay, I got this email that you are invited for the oral interview on 6th September. So that's why I am here.
It is from who, please?
I also
Can you
got the
show-
same email, sir.
Can you show it again so we can see who sent the email?
It's from the university side. It's the official email of the university.
Yeah, but can you move so we can see the sender?
The sender, the sender.
Okay.